Dan, thank you.
All right, so word of the day is peanuts. Okay, so this is, I usually give a yearly talk, so, but Len wanted me to do this 6 months after I gave my last one, so I'm here again. I couldn't even change the year on this one, so it's still 2019, that's the last one I gave. He originally wanted me to do this 3 months ago, so it would have been a 3-month update, but I told him I wanted to do it later in the year, and he said December, and I don't argue with Len when he tells me to do stuff. Okay, I have some disclosures. I get research support from all these different groups, but I'm not a consultant or anything for them, so nothing I'm going to say is going to be. Just to remind everybody, so what was Asthma Inc. for many years has changed its name, mainly because we don't. What is that?
Can somebody please mute their mic?
Mute.
Please mute your mic, please. Thank you. Where our old name used to be Asthma Inc., but we don't do any asthma anymore. We now have no asthma studies. In our last 2 asthma studies, we recruited a grand total of 0 people. So it's, I think, fitting to kind of move on from the past and head forward. But the mission stays the same, to continue collaboration with the folks around here. Is it still going on?
Yeah, it is.
Please, whoever, mute your mic on your other end. We hear you. You must be eating something. I don't know. All right. So to continue our partnership with local groups, and well, I'll talk about it in a moment. One of the things that we're going to be doing very excitingly coming up in the not too distant future. This is our website, seattleallergy.org, where you can kind of, if for anybody, so it's not just us at Northwest Asthma and Allergy, it can be anyone. We take everybody. Although we are in the same building, we are a separate organization. So please, if you have subjects who are, or patients who are interested in participating in clinical trials, or are interested in participating in food allergy trials or other trials, we'll talk about the other things that I have going on. SeattleAllergy.org is something that you can tell patients to go to, and then they can go into our research database. It's all linked up there. I have now well over 1,000+ individuals that have put their information here, which is a wonderful thing for fellows to maybe plunder and look at like kind of prevalent stuff, which we have not done quite yet. So it could be a project in the not too distant future. I'm looking at people in the room. All right. So the other thing that we've kind of formalized and have updated is CSAC, the Seattle Food Allergy Consortium, which is kind of unique. We're very friendly here in the Northwest, much different than the rest of the country, where different organizations can be antagonistic to each other. So this is a collaboration between University of Washington, VM Benaroya, us at Northwest Asthma and Allergy, and at SAARI as well, all coming together with the single focus of collaborating together to help with food allergies. So this is where we tend to be the clinical research arm. The Benaroya guys are doing, and I'll talk about a little bit what they're doing, are the kind of the bench research arm as well. And the next venture that we're going to go into is a new version of FAIR that will be coming up in the not too distant future. We've been a FAIR Center of Excellence, both us as well as Virginia Mason, but this is a new version that's going to kind of take a similar type of venue as COFAR. If you're familiar with that, the NIH kind of collaborative for food allergy. Instead, using slightly different recruitment of sites, where certain regions become a center of centers that will then distribute studies throughout the entire country. And so we're applying for it in the next 2 months or so, and it's a pretty significant grant. For just support, it's $500,000 over a 5-year period that will be split between our 2 groups, and so hopefully bring more and more research to the Pacific Northwest. So let's talk about clinical food allergy research and the stuff that's happening here locally. All right, so just to go over the problem real quick, so estimates of up to 15 million Americans have true food allergy. That's 4% of adults and upwards of 8% of children. This crosses throughout international lines, predominantly in the first world. So Australia as well as Europe, there's similar numbers. Australia's a little bit higher in 10%. Between 1997 and 2011, there was a 50% increase in food allergies. That was reported by the CDC. Top allergens still remain milk, egg, peanut, tree nuts, wheat, soy, fish, and shellfish. The milk and egg thing has always been the case for now well over 100 years, where the very first food allergy that was described at the turn of the 20th century was egg allergy. That was revealed by reactions as well as a relatively new technique of skin testing, just to give you an idea how old skin testing is, that we still use today. But the big difference is that the lack of outgrowth and the severity of it. So whereas it used to be kind of good rule of thumb, most have outgrown it by kindergarten age, we're seeing more like 9, 10 to 15 years of age by the time a lot of these children grow out of it. So, and the propelling of true milk and egg allergy into adulthood is definitely increasing. So estimated cost of about $25 billion annually. All right, so in the Northwest, so 2006, 4,200 office visits with food allergy diagnosis, because this pulled it from our EMR. This was the very first year we used EMR in our system. 2017, this is the last time I pulled it, 12,000 office visits related to, so it was nearly 60% of all incoming new patients that were coming in. Just to see graphically, we kind of see this as well, where we have food allergy, all food allergy, then peanut allergies was tree nut allergy. Definitely has increased over the last decade or so. So one of the key things that we had as one of our arsenals in the food allergy is to truly diagnose food allergy, which will require food challenges. All right, so as you heard last time talking about testing and everything, it's pretty crude. And so unfortunately, we're losing our sponsor over at Children's to continue the food allergy high-risk challenge clinic. So this will no longer be available by the time this summer rolls around. I'm hopeful that there will be. Hello out there, we hear you. All right. So hopefully there will be a new faculty member that will come onto play, that will do what I say, and be able to create a new food challenge clinic that will become much better than the previous incarnation. I'm looking at somebody in the audience. All right, so food allergy confirmation, as I told you, the current gold standard is the double-blind placebo-controlled food challenge, right, which is time and costly. Skin testing, blood testing, component testing, all these things in the absence of history is no better than the coin flip. All right, so you can spend $10,000 worth of testing, but no better than whoever just coughed. Please hit mute. So the absence of history of type 1 mediated reactions is no better than a coin flip. So how many of these positive skin tests are actually not truly allergic? It's a big question, and so we need a better solution. So one of our collaborators here in town, Dr. Wambre, is working on something now to see if we can investigate this and maybe come up with a better solution. And he actually utilizes a lot of the fresh blood samples that we use in our clinical research trials, straight from our patient that's directly transferred over to Benaroya down in downtown Seattle. And so a lot of his work is based off the blood samples from our patients, along with other patients in the clinical trials. And so his big work that came out a few years ago is regarding the Th2A cells, which is a subset of pathogenic Th2 cells that are terminally differentiated, that he deemed the Th2A, which is for allergy. So they're allergen specific to grass, to peanut, to other things, and they have a particular characteristic of being CRTH2 positive, CD61 positive, and CD49D positive. And so when you isolate and use flow, the top flow pattern is an allergic individual, and the bottom is a non-atopic individual, and with a bunch of, and so this represents the Th2A cells, and in this atopic individual, and this is not really present in the non-atopic person, and we can see that also here in a graph format. So this is something that, this is an effector cell that perhaps this is a much better and more finer tool that we can utilize to see if somebody's truly allergic or not. One of the other interesting things is that in the treatment group that was in the AR101 or the Aimmune therapy, he looked at the prevalence of Th2A cells at the beginning, so pre-OIT, and the two different groups, placebo versus active, and there was a cell-specific, all right, so peanut-specific drop in the Th2A cells. It looked like that. This is how oral immunotherapy works, by selectively deleting these cells. And so it can help guide us for a lot of things. So if, for instance, in patients who were on the multi-food therapy, there were some patients who had extraordinary responses, almost like their skin tests would go negative, their blood tests would go negative, and were for certain things, but other things that were still positive. And so some, they could say they were cured, other ones not so much. So this could definitely help us, kind of guide us in terms of utilizing it as a much more improved test. So can this answer all the questions that our patients routinely ask us when you get a positive test? How allergic is my child? How worried should I be? Is my child really allergic? So right now, the best that we can do is, let me give you a little bit of this stuff and see if you react or not. And those are time-consuming, costly, it's expensive to do challenges. If they have a couple of hours to kill, fantastic. And a lot of very motivated parents and children are ready to go do this. I have this one child that I'm working through right now. We've gone through about 12 total hours worth of challenges at this point, all of which had positive tests. So he had, like, for instance, he had a hazelnut with a 12-millimeter wheel, a 15 immunocap, absolutely no reaction whatsoever. And the same thing was true for almond, as well as other things. We were just going through it, and they were at that transition point of, he's going to go to college in a couple years. Do I have to worry about this? Do I have to do an EPIT? So we're going down by one, and even with these positive tests, he's absolutely no reaction whatsoever. So could this type of technique help? And so that's part of our collaboration that we're doing with Benaroya, is to work on this type of stuff so that we can have the challenge data plus the Th2A cell prevalence, along with other things, and through immunotherapy, to see if we can answer these questions in a much more effective way than what we currently have available. All happening here. Okay, so let's talk about the stuff that is being investigated right now for the current food allergy treatments, and we'll go through each one of these. There's some that are very close to FDA approval, other ones that are a little bit further away, but I think 2020 will be the year where we're going to have our first ones. So epicutaneous, oral gene therapy, oral immunotherapy, plus a biological modifier, subcutaneous, as well as peptide immunotherapy. All right, so we're going through these phase 3 studies. So right now, the current phase 3 studies that have been completed are the peanut patch and the low-dose peanut OIT. So let's talk about the peanut patch first. We did our first foray into the food allergy treatment world was with Steve Tillis back in 2013, where we got involved with the VIPE study in the phase 2, and we're currently still doing the PEPITS in people, which is a rollover, and that's being extended into 5 years of ongoing therapy, and as well as the safety peanut study called Realize that will be ending soon. All right, and this is what the patch looks like in a previous incarnation. There's actually like a little tegaderm, that like material that kind of wraps around it, that kind of helps seal this to their back. All right, so this was published in JAMA a little bit ago, multicenter, and this is basically how it goes. So it goes on the child's back, all right, and so this is the mid-back, and they go through this rotation where they start at number 1, work down, all right, and how the dose escalation basically happens is with time. So at the very start of it, it's applied for 2 hours to 6 hours, depending on the protocol, and then weekly it's increased a number of hours till eventually it gets to a 24-hour period, usually by the time a month rolls around. And so they place the patch for 1 full day, and then they remove the patch and they put one on the level below it, and so on and so forth, with a rotation all the way around. And this is the basic setup. So there was 31 international sites. There was 218 boys and 138 girls. Median age was 7 years of age. All right, there was 356 children randomized, with 238 in the patch and 118 in placebo. Qualifications, they had to have a peanut IgE of greater than 0.7, with skin testing above 6, or above 8, depending on their age. And then they had to go through a confirmed double-blind, placebo-controlled food challenge. This was a pretty strict food challenge, so they needed to have pure objective signs and symptoms. So if somebody was just nauseous, it wasn't enough to stop it. If they had a single hive, it wasn't enough to stop it. They needed to have moderate to severe symptoms in order to stop it. So this is a pretty high bar for them to reach before we would stop it. And so it was a standard practical guideline, so 1, 3, 10, 30, 100, 300, and they had to react at or below this level. And so to be included, they need to have an eliciting dose of less than 300. One of the more important things to look at between the two studies is that the difference between eliciting dose as well as reacting dose, and so we'll talk about that in a little bit. So depending on what their eliciting dose, what level of reaction that they had, so 1, 3, or 10, they needed to be able to tolerate over 300 milligrams. And if they got to a high eliciting dose, 30, 100, or 300, they need to reach a total of greater than 1,000 milligrams at their exit challenge. Just to give you a perspective, a typical peanut is about 280 to 300 milligrams. So they had to be able to tolerate a peanut or up to upwards of 3 peanuts. All right. So one of the statistical requirements for a positive trial for this, the FDA mandated that a margin of 15% or greater was to be used as a lower criteria for the 95% confidence interval to determine if this was a positive trial result. The 15% margin was based on historical allergy studies, but never with a food allergy study. So this was just a number that they picked. All right. So this is important. So one of the things, so this is a response rate. So for the peanut patch, 35% or 84 subjects reached the primary end goal, and there was 13.6% or 16 in the placebo arm did this, with a difference of 21%, which was statistically significant. But if you look here, 12.4 was the 95th confidence interval. So unfortunately, this did not. This difference was. Did not meet the permitted 15% the FDA mandated for a positive trial. So it did not meet the statistically study for success per the FDA. Although the difference was statistically significant, but not the 95th confidence interval. The secondary points, so cumulative reactive dose is the total amount of peanut consumed at the end of a challenge. And so the mean cumulative reactive dose was approximately 210 milligrams in both groups at baseline. And then in the active treatment, they were able to tolerate 900 milligrams, or a median of 444. In the placebo, a mean of 360 with a median of 144 milligrams. Why so many placebo kids got better on this is still a mystery, but a pretty significant amount did better with just doing this. So especially with the strict criteria that they had, I'm truly puzzled how it had that how that occurred. In most of the kids that we had that were placebo, just looking back on it, they actually reacted at a lower dose, not a higher dose. So which is kind of more typical what you would see. And actually, some of the work that Dr. Wombre has done looked at Th2A cells after the initiation, after challenges, and they proliferated and they were increased amounts in circulation. So, which makes sense. You kind of stimulate something, expose somebody to something, perhaps that haven't been exposed to many, many years. Of course, you're going to get an immune response, and so they tend to be more sensitive. So it really surprised me that this is how it was, but that's what happens. That's why you do the work. So a total of 89.9% of the patients completed the trial. Treatment adherence was 98.5%, so it was pretty well tolerated. So only 4 out of 238 in the treatment arm stopped due to treatment-related reactions. And most treatment-related reactions were all related to the site. So site itchiness, redness, and swelling are the most common, and that just diminishes over time. They all complain about this, especially at the beginning, but after a couple of months, this truly does diminish. And topical steroids is usually enough to take care of it. One of the more important things is not to come back around when you're actually doing this onto inflamed skin, because that could lead to scarring. In the VIPE study, there was a few kids who weren't really compliant with the treatment of it, and so they had these scars, circular scars from the patch. One kid had it tattooed because he got so much better afterwards. This was an older kid, teenager. But most of the time, if just plain old hydrocortisone doesn't work, triamcinolone takes care of this. So like a moderate potent steroid, and like I said, it gets better with time. There were 26 cases of anaphylaxis reported in the study, 10 cases in the peanut patch group felt but to be possibly related to the patch therapy, so to a total of 8 out of the 238. None of the reactions were severe, and 5 out of the 8 completed the study. So currently, what's going on right now with the patches, we're doing the toddler study. I see my coordinator for the toddler study right here, and it's in full swing right now. They're having a lot of trouble filling up the study because of the amount of screen fails that are happening. So these are kids with positive immunocaps, positive blood tests, positive skin tests, and who have possibly a history of it that pass challenges. So it's now the screen fail is about 50%. So a lot of people don't want to hurt the kid, all right? Don't want to challenge the toddler. But evidently from this, I think getting some of this interesting screen fail data might be interesting to see, like, what truly is the prevalence of this based on the testing that we're doing. So the peanut patch initially had their NDA that was initially put in October of 2018, but it was pulled due to the FDA questions of manufacturing and QC, so they didn't want to get rejected. So they pulled it back, and they've resubmitted it, and it has been accepted in October 2019. So we'll see what 2020 brings, whether this will be an option. There's been some, despite the not meeting 95th confidence interval, there's some, just as a kind of back talk about this, that there's some favoritism towards this because of the safety profile and as another option for subjects as well, for patients as well. All right, so the peanut oral immunotherapy trial is the other thing that we've done. So the first foray into this was the Palisade trial, which is that has been completed, as well as the Palisade. Now there's this long-term rollover extension that's going on right now. It's called ARC-08 that all the subjects are in now. So some of these children have been in this trial for years, like 2 years at this point, doing the daily dose. All right, and so the target dose for the Palisade study, and we'll talk about the logistics in a moment, is 300 milligrams, so about a peanut's worth. Ages range from 4 to 55 years of age, skin prick above 3, anemic cap 0.35, and they needed to have did a double-blind placebo-controlled trial. Primary outcome is in clinical reactivity to get above 300 milligrams. All right, oops, see, all right, there we go. So essentially, this, after they get through the double-blind placebo-controlled screen, they start with the initial dose escalation that gets up to upwards of about 12 milligrams total in a day of peanut protein, and then they do that their first day, and then they come back and they start at 1 milligram, and they do that daily. This is all kind of contained inside this little capsule that they open it up. So oftentimes you'll hear in the media that there's a peanut pill. This is not really a peanut pill. So the pill is just a container device for the powder. The powder is then put into something mixed that's cold, so usually applesauce, pudding, or something else that the kids like, and then consumed on a daily basis. So this is not like a pill that they can just swallow in, all right? And then it's slowly built up over every 2 weeks, depending on their tolerability, and that goes up to 3, 6, 12, 20, 40, 80, 120, 160, 200, 240, 300. This is every 2 week intervals going up here, assuming that they do very well. So if they're having increased GI side effects or they had reactions, then it can be extended. So anywhere between 6 to 9 months is the kind of the build-up time it would take. And then every 2 weeks they're coming into the office for the so-called up dose. So this was the Palisade publication in the New England Journal. So over here we have in light blue is the placebo guys versus the ones in AR101. So this is people tolerating at a at a reactive dose of 300 milligrams. So they didn't just have 300 milligrams. They had approximately 440 milligrams at this. So because it's a challenge, so it's the doses are built up, and so over 75, almost 80% tolerated this level, and then the primary endpoint here was able to tolerate greater than 600 milligrams, and so which was about 900 milligrams of peanut protein, and they did quite well compared to placebo, and then those who are able to tolerate over 1,000 milligrams, also significantly different than placebo. The other, let's see, yeah, there we go. The other interesting thing is the relative severity that was seen. So this was at the exit screening. So this is placebo, and this is AR101. And these are the symptoms that they had. So yellow is moderate, green is mild, and red is severe symptoms. And so you can see at the relative doses that they reacted to that the group in the placebo arm, whilst a lot of people had these mild reactions, they had much more severe reactions in the placebo arm versus the treatment arm. So while they did react, their reactions tended to be mild, and I also saw that clinically as well. So study discontinuation, withdrawals due to AEs in the treatment arm was about 30 or 8%. And then 12%, not due to AEs, I'm sorry, 8%, and then due to AEs was 12.4%, and then the predominant reason is GI. So GI side effects are definitely a concern for this, especially with the concerns of leading to eosinophilic esophagitis and other symptoms that are kind of like that. What most of the time you see is not usually immediate GI reactions, it's delayed. So they'll feel nauseous or more vomity like 12 hours later. It's really it's different than what you would typically associate with an immediate type reaction. They definitely have this as a concern. And so it's something that if you continue doing that, it doesn't get better. This is something that persists and continues and worsens. So it's something you have to really focus on. And it is definitely something when my kids come in for their evaluation, I constantly ask them about how's your stomach doing? How are you feeling? How's your abdominal pain? How's your appetite? Do you have a loss of appetite? And I use the vital sign of weight loss. So if I see weight loss in a kid, these are all reasons to stop because they're not eating as much. And so we need to be very careful. There was about 3 or 4 of our patients out of the about 20 or so that did develop EOE actually, at least at some point during their treatment process. So this is definitely a real thing we have to really consider when we're doing this. All right, and then true reactions definitely did occur, and so I definitely saw this for sure, where giving over the phone epinephrine when they called me emergently in the middle of the night is definitely a real thing. That's a good trick. All right, so where it is, so it's cleared the very first hurdle for FDA approval. Not without some controversy, but they're definitely progressing. So more than likely, within the next year or so, there'll be 2 products that will be approved for peanut allergy. This being one of them, probably earlier, and maybe the patch. What's next for Aimmune is egg OIT, which is currently undergoing right now. This is both people who tolerate baked egg as well as non-baked egg tolerant folks, and we're a site for it, and we're recruiting right now. So let's talk about a little bit of the logistics of all this stuff, since probably before the next time I talk to you guys, this will be something that might be available and for you to think about in terms of treatment. So let's talk about these practical considerations of this. All right, so logistics of AR101 OIT. This is every 2 weeks they come into the office for 6+ months with observation, and they must follow this dose escalation process. So on average, I would say 7 to 9 months is usually, 6 months is like if they're perfect, they have no symptoms whatsoever and they breeze right on through. With the Viaskin Peanut, the EPIT, there's no specific interval. My kind of recommendation is you check on them after a month and 3 months, and then it's every 6 months. So it's really infrequent in terms of it. It's pretty easy and pretty well tolerated stuff. After hours reactions, you will get calls when you're on when you're on OIT. So you have patients on OIT, so you will get these acute calls. Most of this is around like illnesses. That's the predominant thing. So these are children, so of course they're going to get sick in the cold and flu season. Last week I got like 10 texts and messages about from 2 families about kids throwing up, vomiting, flu-like symptoms. Do I take my dose? Do I skip my dose? What do I do? What do I do? What do I do? Yeah, that was last week. Rare, if any, calls for the Viaskin stuff. The only time I've ever gotten a call was one time because it was very beginning and the person, the child touched the patch and then they touched their eye. And so I told them not to do that anymore. And had to talk to the urgent care doctor for not giving them epinephrine because I had to convince them it wasn't systemic. It was a local reaction because they physically put it on their eye. The age range is a little bit different between the two. So the likely approval for the Viaskin is going to be 4 to 11 years of age, and the AR101, it was 4 to 17. I mentioned they went up all the way up to 55, but since the majority of children, majority of adults don't care about their peanut allergy, it was very difficult to recruit, get a significant number for it. So one of the things you're going to find is they kind of go doing these adult trials is that they are like, yeah, I'll have to stay away from it. I don't have to worry about it. That's usually kind of how the attitude comes. But when you have the hyper nervous mom, they're going to be participating in trials or they're doing this. Precautions. So AR1, there's quite a few. You need to avoid anything that will raise your body temperature, particularly during exercise. So hot baths, hot tubs, all this stuff can increase reactivity during this time. So one of the worst reactions that I had was somebody on AR101. They took their dose, and they immediately went outside to play ultimate frisbee, and they were at camp somewhere on the Olympic Peninsula. I was a 2-epi kid. I was talking to the mom through her cell phone, who was talking to the counselor that was over there. So it was a nice little phone tag I was playing out there, and they took him to the hospital. The restrictions, right now it's an hour prior to taking your dose and to 3 hours post. So if you have anybody who's interested in some sort of club sport activity or high-level activity like the teenagers, we have a very fit group here in the Pacific Northwest who want to do all sorts of sports. Fitting in time to do this, they have their morning practice, their evening practice. They just don't have time to fit this in their schedule. And exercising definitely does cause reactions. NSAIDs need to be thought of as be thoughtful with its use. So if you are sick and you take NSAIDs and you take your dose, more likely to have reaction. For the EPIT, just take it off prior to swimming if you want to. Local dermatitis reactions with topical corticosteroids is pretty much the extent of what you have to watch out for. I did have one kid though had an interesting reaction when they sweat. They would fill up the patch and then the peanut in it would actually kind of leak around it, and so they would get hives kind of around the patch. So we just took it off before they went and did exercise. They were a really sweaty kid. All right, so AR101, most of the patients do reach maintenance dose, and you do know that they're desensitized. So you do know that they will be able to tolerate at least 300 milligrams. That's no doubt because they're doing that every single day. And so, which from the study certainly has offered them accidental exposure. So this is certainly an advantage of the AR101. But for the EPIT, you don't really know until you do a challenge. So there's no real way to know that you're doing anything because the majority of kids in that first year really didn't respond. So what are they going to do after 2 years or 3 years? Good question. So you have to find that out right now. And so I'm doing in about 30 minutes from now a challenge on somebody who's done the without a pre-challenge to see how well they do off the patch. So it does require challenges to figure this out. So.
Do you have any data on component testing in these people along the way? Or
Yes.
Is this just all clinical?
There's an elaborate amount of information about all these patients. So they were characterized they have IgG4, IgE, and components for each one. I think it's probably in the papers. I don't have it for you. But yes.
Does that predict anything, or is that all just data?
Did not seem to predict anything. Yeah, so component testing is something I use with a definitely with a limited thing, especially since how much it costs. So disadvantages, as I mentioned, the Q2 weeks, the possible anaphylaxis for the AR101, the GI side effects, upwards of 15 to 20% do not remain on therapy after you complete it. That's one of the big problems with this stuff is it's not exactly appetizing. And a lot of them, especially as they get a little bit older, they do have this aversion to peanut itself. Kind of makes sense if there's something that you react to or it makes you feel terrible. Do you really want to eat it every day? Just think about this. Like, think about a food that you hate. And this food that you hate causes bad reactions in you. Now, a little bit of it every day. Does that sound like a wonderful thing to do? It all depends. So some people don't care about it. Other people do. So it's definitely dependent on the child. So there are some children who are nonplussed by it, and this works out well for them. But there's other ones that you could see that they're just not going to tolerate this at all. And the moment that they can stop, they stop it. Viaskin, upwards of a third, have no benefit in the first year. Efficacy is unknown until you do your challenges, and then you have your side effects with the EPIT. So that's the current state of affairs. What the logistics is, out of curiosity, anybody who in this room is going to be doing OIT or when this FDA product comes out? Is anybody raise your hand? Any allergists in the office? No. Yeah, maybe. Okay. So definitely. So just right now, there's a couple logistical hurdles to get over. So as I mentioned to you, the Q2 week visits. All right. And so right now, there's no set up billing codes for this. So the observation time is an hour and a half for this, that somebody's going to be taking up a room. And you can't just throw them out in your waiting room because this is children. So, and you're going to do this every 2 weeks for the good 6 to 9 months. So how are you practically going to do this? And so there's going to be a lot of logistical hurdles, also payment and compensation. So one of the things that Steve Tillis, who's joined Aimmune, that's one of his jobs right now is to figure out those logistical hurdles. The other big thing is that the AR1 product is refrigerated. So you can't just go to Walgreens and pick this stuff up. So this has to be specialty pharmacy based. And so anybody who's ever dealt with a specialty pharmacy, they suck. And so you can't not get this dose for 3 days, 4 days as they're waiting for their shipment to come in. They have to get it now. And so, okay, so you're gonna have samples in the office. Well, am I gonna have to buy 5 different refrigerators to house all the samples? Because they're not just for some of it, you have to have for every single dose. So again, logistics for this to kind of be overcome. So while this will be approved in the next year or so, I think the logistics of handling this stuff is going to be something that will take some more time before it actually kind of becomes a more routine thing. And picking out who will be the good candidates. Based on my experience, I think about 25 to 30% of peanut allergic would be candidates for this type of thing. So this is definitely not going to be 100% of every peanut allergic that's going to be doing this. So usually when people come to me and they have a special consultation, I want to talk about food immunotherapy. Okay. And I talk about the logistics. I talk about the QT visits. I talk about like the risk reaction. I talk about the restrictions on activity. And I see that, oh, nah. All right, okay. We'll see if something's better. So can there be something better? And that's what some of these other trials are going on right now. So this is Mark 1. So this is a good place to start, but maybe there's better things to come. So Xolair and food allergy, this is something that's been going on for a while now. We've been part of the multi-food study with Stanford several years ago to wonderful effect. I'm doing a few patients I have on this that I'm collaborating with Ben Arroyo that are going through this really easily. Just to give you a sense of this, you basically use Xolair as with the asthma dosing. So you use a table for the asthma dosing, and you can treat them for about 2 to 3 months with just Xolair alone. And from the previous studies, we saw that there was between at most a 60-fold improvement of their tolerance of peanut. So people who reacted at 5 are now able to tolerate 300 by just doing Xolair alone. All right. So I've taken a couple of patients and done this, where I've given them Xolair, and then in one day I reach the same maintenance dose as Aimmune in a single afternoon or single morning. All right? Sounds a little bit easier, huh? And they do really, really well. No reactions. Nothing that happens. So it's a really interesting thing. So right now, one of the big problems with this is that it's not something that's covered currently. It's not approved for this indication. So COFAR is currently doing a study with this where they're looking at just Xolair alone, seeing this kind of improvement versus placebo, and then doing Xolair with multi-food immunotherapy that's recruiting right now, with the hope in the next year or so that they're going to have some positive results and maybe get this as an indication. So this could definitely turn something, it's rush, so you could do rush, just like people do cluster immunotherapy. This is a way to kind of take away all of that Q2 week stuff and be able to like jump on this. The other great thing about this is that you don't have to just be relegated to one thing. You can do multiple foods. So in the multi-food study, we did 5 different foods simultaneously. So there's a lot of advantage to this type of therapy going forward. And so especially if this becomes a covered thing, I think this would be definitely a way forward. And having standardized type foods would be a great asset to have. So there's definitely a role for Aimmune, because measuring out 5 milligrams, 30 milligrams is a real pain in the tail. So having something that's like preset that you can do this for patients, and being on this like set formulation is definitely advantageous to have multiple types of allergens. The other thing that we're doing right now is the Aimmune Regeneron study. To both Regeneron and as well as to Aimmune's credit, is that despite having something that's on the way to FDA approval, they decided to, like, you know what, we're going to do this for science, and let us take a look at, see what happens when we add dupilumab to this regimen. So it's AR101 with and without dupilumab, and it's a phase 2 study for ages 6 to 17, and we are actively recruiting that as well as in Seattle as well, all right? And so IL-4, IL-13, this is what's being blocked by dupilumab, and that causes interference with generation of IgE, eosinophils, T cell, mast cell, multiple different pathways are being blocked here. And one of the other interesting things that dupilumab is being investigated for right now is EOE. And as I mentioned to you, EOE is one of my biggest concerns when it comes to food oral immunotherapy. So maybe this could abrogate that and prevent that from, yes?
Do these patients need to continue with biologics after they've been?
So I don't know, for this one, we don't know, because we're actively doing that for that one, but for the Xolair, no. So it stops. So you do it for like 2 to 4 months. So, which depending on what protocol you use, and then you stop using the biologic. So then you kind of take over with it. So it kind of gets you built up really quickly, and then you kind of just use the standard oral immunotherapy. Then it becomes much more practical at that point. So you can do like peanut itself, and do 1 peanut, 2 peanut, 3 peanut, that type of thing as you kind of build up. So you're not doing the micro amounts that require from just a straight OIT. So this will be an interesting thing just to see what the side effect profile will be. And that's, like I said, ongoing right now. A little bit tricky to recruit for. This is shots every 2 weeks for an entire year. So, kids are not super down with it. But if you have any interested parties, we are definitely active recruiting. We're happy to take them.
Dan, and when does the OIT start in the?
So that's only, so they do that
3
for 2 weeks prior. Yeah.
So just 2 weeks.
4 weeks, sorry. 4 weeks. So they do 2 rounds before they get, so they get loaded, and then they get, so they get their, depending on their weight, 600 or 400 milligram load, and then they do their 300, and then the following one is when they start their oral immunotherapy. And it's exactly the same thing. And as a poll for that study, everybody gets OIT. So it's 100%. So that's not placebo controlled. The only thing that's placebo controlled is the dupilumab. Yeah.
So if you look at the data on people that are on Xolair or dupilumab for other diseases, do they stop anaphylaxing?
No, they don't necessarily do that. So there was some thought that maybe the kind of the hive dosing would be a way to like use this as treatment, as like kind of induction, some type of induction, and that did not work out. So Stanford did publish, like they had a paper, a poster at the college meeting, and it just did not work out. So the 300 milligram Q monthly was just not enough. So it really required the asthma dosing. The problem with the asthmatics is that there's a reason why they're on Xolair, because they're pretty severe asthmatics, and so those are people that have been excluded from almost all these studies, and so we don't really have great data on those folks because you kind of don't want to do this on somebody with that fragile of an asthma type control. But yes, it's a good question. But those people that would be on the asthma dosing would be disqualified from even participating in this type of thing. So they had to have mild to moderate asthma for sure. And it's definitely different than like the idiopathic anaphylaxis because I've used the 300 milligram dosing in those folks, and they work really well. It works really well. The chronic high dosing, and so it's very different. So with this kind of rush immunotherapy. And so with this, you're also doing food on top of it. So you're kind of having both aspects. That's kind of continuing on it. So there was another company that's been working on a subcutaneous peanut immunotherapy. Peanut immunotherapy, SCIT peanut immunotherapy has a scary nomer from the 90s where a subject that was at a National Jewish trial died. It was due to a pharmacy error rather than related to the SCIT itself. But that kind of killed food immunotherapy for almost 20 years. So it really kind of put the brakes on everything. So people were doing food immunotherapy back in the 90s. C. Tillis was started as a fellow the year after that happened, and so he would be at bedside giving people shots of this stuff and staying with them the entire time to make sure that they were okay. So there was a lot of baggage that came with this. Actually, some people in my practice did not want to be sub-investigators because they didn't want to be involved with this at all because of the risk involved. And the company took it in great seriousness. So these people were given their shots and they sat in a room for 4 hours afterwards, and it was weekly. So weekly shots for 4 hours. With observation. And so they've completed their 1B. This was in peanut allergic children. In our cohorts, they tolerated well, but the results are pending. So they were supposed to presented at YACI earlier this year, but they didn't. So we'll see how that goes and what's going forward. And this is a aluminum hydroxide absorbed peanut extract as opposed to just raw peanut. So it was a little bit attenuated. And this HAL allergy is something that we're not super familiar with in the United States, but they're a very old company that's been doing this in Europe with alum-based immunotherapies with for conventional SCIT for many years. Peptide immunotherapy is a very exciting one. This is something that's coming out of from Australia, a company called Aravax, and they have this peanut peptide vaccine called PVX-108, and this is monthly intradermal injection. So these are peptides of different different components of the peanut protein, and they completed a phase 1 trial. This was just a safety trial in peanut allergic adults, so 1B, and it was found to be safe, produced no anaphylaxis. There was in that trial over 60% had a history of severe reactions to it, none of which reacted. There was 30% that had significant asthma, none of them reacted in anaphylaxis. It was all local side effects, local injection-related side effects. So they're proceeding with a phase 2 study that will be international. So we're going to be a site for it coming up in 2020. Really kind of excited about this one. Peptide immunotherapy has kind of fallen by the wayside, but this seems like something that could be very potential as a very interesting thing. And maybe get around with a lot of the oral kind of aversions that a lot of people have. Also, I kind of like it to be able to control things. It's kind of strange to me. It's like doing SCIT at home, and just trusting somebody's going to do this. That's what we're basically doing with much more severe allergen. When we do oral immunotherapy at home, this will be a way to kind of control that as well. All right, so as I mentioned to you, we're kind of branching out besides other areas of food allergy research. So besides food allergy, we've started our very first hereditary angioedema study. This is a really cool drug. This is a oral kallikrein inhibitor that's actually been submitted for a new drug application, and we are also actively recruiting that. So if you have a type 1 or 2 HAE patient who's 12 and above, we're interested in them. They can be on therapy. So they can be on Cinryze, they can be on Takhzyro, they can be on anything, and they can participate in this. This is an all-comers, this is an open label. They don't have to be on anything This is not placebo-controlled. Pretty well tolerated stuff, apart from some GI symptoms that usually go away after the first month. Very interesting drug and a kind of brand new kind of way of small molecule introduction of this. And then hopefully by 2020, this will be something that will be available to our HAE patients. Not lab normal folks though. So it has to be true type 1, type 2. We're also just got initiated on a Church-Strauss or EGPA study that's looking at benralizumab versus mepolizumab. So if you have subjects who are mepolizumab naive, so that haven't gotten it or have been having trouble getting mepolizumab, and they're on daily steroids, we have access for them to get either one of these drugs. Not placebo-controlled, it's a head-to-head study. So if anybody has these, yes?
What's your age?
Age is 18 and above. Yeah. Sorry. And then the last one that's likely to be coming to us is at EOE with dupilumab, and this is adolescents and adults. And we are kind of in negotiations right now. It's mostly due to like figuring out who my collaborators will be because I can't scope people. But I'm pretty excited about this one actually, to have an actual option for patients with EOE. The phase 2 data from adults looked really promising. It almost got FDA approved based on the phase 2 data. So very excited about this as a treatment option for people as well. So we shall see, and that's placebo controlled. That's 12 and above. So this is all the stuff that we're recruiting for right now. Please, if you have subjects who are interested in participating, seattleallergy.org, come on by. We are happy to take you. I promise we won't steal your patient. We'll send them right on back to you. That's kind of my tagline. If they're asking me questions, go see your regular allergist. All right, so I don't do this alone. We have wonderful sub-investigators. Both Dr. Winteroth and Dr. Sagetto have been wonderful help and been spending more of their time with us, being sub-investigators on our study, and along with the Seattle docs, so Dr. Allman, Lasley, Tatum, and of course Dr. Brunt here. And our staff has been wonderful. Lisa Miller and Kim Varner are our managers, and we have two of our folks here in the room right now who have been coming to this and learning more about allergies, which is wonderful. So both Ramon and Catherine. And so this, and along with all the study participants and parents for giving their time to us and like participating, couldn't do without them. All right, as I said, the word of the day is peanuts. Do we have any questions? Yeah.
What happened with the study in England where they just had the parents increasing the oral dose of peanut powder over a span of years? Has that just died, or is that?
Oh, you mean like the LEAP study? Is that what you're referring to?
No, it's actually just the oral powder, and they just dispensed it to the patients, and they actually updosed their kids at home.
Well, I mean, it all depends on how allergic the child is. That's really what you kind of determine. And so I actually do that as well. So I threshold test people, and so I'll do low dose exposure challenges where I'll do it clinically, and I'll do a similar type of thing where we're doing here. And if they can tolerate a practical amount of peanut, where it's basically where somebody could reasonably get at this without being too burdensome. So I use candies in that case. I use like Reese's Pieces and or Reese's peanut butter cups and things like that, where you can cut them in quarters. Or there's about 70 milligrams of peanut protein in a Reese's Pieces. And so if somebody tolerates over 400 milligrams or at 400 milligrams they have a reaction, then you can do 25% of that and use that, and then we just kind of use that as low dose immunotherapy and just slowly build them up over time without having this kind of more organized things. This type of thing is really for the people who react at the very low doses. So like, for instance, like when we're doing challenges, I don't really super pay attention until we get to about 30 to 100. So most kids react around 40 milligrams. That's typically what I'll see, and that's usually kind of like where we're at. When we're doing our kind of like rule out challenges, we usually start like a peanut, right? Like, peanut at least. So these are kids that are reacting that much lower, and so you can't really do good like kind of home dose things without having a really good scale and measurements. The other stuff is that there's huge batch-to-batch variability with the AirH2 as well as the other allergic components in natural products. So if you don't have a good safety margin, some of this stuff can be 100%, 1,000% greater between like one bag of peanut flour versus another bag of peanut flour. It can be significantly different. So Bob Wood had some research on this as well. So you have to be very careful. So when you're talking about 1 milligram, 3 milligrams, that can make a huge difference, all right? But if you have a threshold of like, 3-4 times, I think you can be okay by doing this and having good batch-to-batch variability. And if you buy 1 stack of peanut flour, it's going to last you for like 2 years, so so it'll be good for a while, all right? Yeah. Yeah, Frank.
And are any of the companies looking at microbiome modulation, for lack of a better description, to try to make more permanent tolerance?
So there was a Australian study where they did look at microbiome. The only problem with it is that they didn't do challenges before. Yeah. They did challenges after, which is basically meaningless to me. So I wish they designed this. It was a powered study. They just designed it poorly, and they didn't challenge them before they did it. So, as far as I'm aware, there's been no good large number study looking at microbiome and then what microbiome and what do you do. There's been some interesting work from like Sinai with Chinese herbs, but again, just like practical issues. And it's a natural product, so it could also be batch-to-batch variability there too. That's the other thing I worry about herbal medicines, is that it could be tremendously different depending on like what side of the mountain that they grow on, like sunny versus shady. It can be hugely different. So which is why there definitely is a role for things like Aimmune, where you know exactly how much ARH2, 3, and 6 is in it versus some sort of natural product. So it's not just peanut flour. This is like specifically measured out stuff, and so that's why it's tricky and complicated for this. So to kind of figure this stuff out, so they need to know how much of this protein is in this food substance. So that's what's taking some time for these other things. You can't just throw it out there. I was chatting with somebody from the USDA who was developing like the assays to measure specifically amounts of in things like walnuts and cashews and other things, so that they can start doing that. And that, I think, is the next big area. Once peanuts kind of get settled, I think the tree nuts, specifically cashews and walnuts, are going to be the next big foray in the nut world, along with the milk and egg, which is kind of like the biggest concerns right now for the younger set.
Right. Well, I've had the same experience talking to parents that ask about it, and you start talking about the updosing and the laborious nature of it, and almost always it doesn't create permanent tolerance.
Yeah.
They sort of, their enthusiasm drains a bit.
Yes.
And I said, but we'll offer it, we'll talk about it.
Yeah. So does it mean it's gone away? No.
Yeah,
So
no.
this, as I didn't emphasize this, but it's all desensitization. This is not tolerance. This is not treatment. Now, what will happen after 3-5 years of therapy? They will do something, but then they have to stick with it for 3 to 5 years. Same thing every day. Every day. All right. Anything else? All right. Well, thank you very much.
Connie's like,